I think that really reinforces a message we got from Sheridan yesterday by the pool, but also this morning from Dwayne, that the way we interact and engage with TOs and rights holders, as Adrian said, is evolving and will continue to do so under NESP too. So some important messages there. We're going to change tack a little bit here. We're moving away from discussions about management and Indigenous tourism and Indigenous aspirations, and we're going to hear from Kate Quigley from AIMS about the genetic traits of corals that survived recent bleaching events. So looking now at some of the NESP research. Thanks, Kate.
Hopefully we can use a bit of Dwayne's time as well. I'll try to make it quick. So first, I'd just like to acknowledge the traditional owners, past, present, and emerging, especially the Bindle people where the AIMS headquarters is located, and especially Llama Llama traditional owners, which with their consent and really participation, we were able to collect a majority of the samples, especially in the far north, that I'll be talking about today. So I'll just introduce the team up at AIMS, so Lena Bay, Patrick Laffey, Blake Ramsby, and Veronique Moisian. And this project really came about because of the 2016 bleaching event, probably very similar to the response that we heard about yesterday with the mangroves up in the Gulf. And so, yeah, I hope in the years to come, maybe I'll be speaking with other stakeholders on this stage like we saw yesterday. So coral bleaching, I probably don't have to convince this audience that it's a problem on the Great Barrier Reef, and the ARC Center of Excellence has done a really good job documenting the impacts both in bleaching and mortality in 2016, 2017, and again, unfortunately, in 2020. And particularly concerning is that these increased water temperatures do not seem to be going away, and potentially bleaching will become something that's more of a persistent feature of summertime on the Great Barrier Reef. And so kind of just to give a quick overview of what bleaching actually is, we have a branching coral here, and on this side we can see kind of a normal pigmentation. And that pigmentation is due to this really fundamental symbiosis between Symbiodiniaceae, which are small microscopic cells that live within the tissue of corals, and they really provide a majority of the nutrition needed for corals via photosynthesis. And the problem arises when water temperatures become too hot and they stay hot for too long, in conjunction with other factors like high light and potentially disease. And essentially, the symbiosis breaks down. And if these conditions stay like that for too long, the coral animal can die, and essentially it will turn white, so that's the loss of those symbionts, and then if it dies, it's kind of colonized by this filamentous algae. So it was thought maybe about 20 years ago, that bleaching could provide an ecological opportunity for corals to contend with changing environmental conditions. And this was termed the adaptive bleaching hypothesis. So potentially an ecological opportunity for corals to use their symbiont communities to change and respond to future stress. And so if we kind of take this little toy example, corals can essentially, through time, maybe there's some environmental perturbation that occurs, and they can shuffle the relative abundance of those symbiont communities inside their tissues. Alternatively, they can do something called switching. And so that means that they can take up symbionts from the environment, so free living forms, potentially more thermally tolerant types, and again, change their communities through time to essentially have a new phenotype, so respond to environmental stress in a different way. But that really wasn't known how pervasive that these two features are throughout coral communities. So we know from a number of different meta-analyses that that species are changing in response to climate change, and these can tell us a lot about population growth, about colonization events as different species try to move into new habitats to contend with stress, and probably most concerning any kind of local extinction events that we can see. And in the last few years, there's been a number of really nice papers to show that mass bleaching can restructure fish communities. It can actually impact the coral animals themselves, but we really don't know much about what's going on with this very fundamental populations living inside of corals, as well as in the environment in terms of how they would respond to stress. So that was kind of one of the main aims of this NEST project. So, boop. So everybody kind of had that really nice diagram of paddock to reef yesterday, so I was thinking, you know, what's kind of a similar conceptual diagram that I could use, and really understanding what's going on with healthy reefs and how they respond to bleaching is so important, because even if bleaching becomes a persistent feature or it continues to happen, as long as we have some healthy reefs, we can really the ecosystem can recover due to reseeding events, whether that's due to natural spawning events or to the reseeding through interventions such as restoration. And so, you know, kind of sharing the adaptation, whether that's a genetic adaptation from those healthy populations or through their symbionts, is really a way to incorporate resiliency into management. So to do this, we needed a long-term data set, and so luckily Ames is very good about organizing their tissue samples, and so I dug into the Ames long-term tissue storage and found a number, thousands of samples that we could go into, spanning from 2003 up into 2016. And then we supplemented new samples using either the Ames boats or partnerships with Great Barrier Reef Legacy to collect post-bleaching samples, so samples after 2016, 2017, up to 2019. And then we were able to really collect across essentially 26 different reefs, both in the far north of the Great Barrier Reef as well as in the central Great Barrier Reef, to kind of bring together this data set of pre-tissue samples as well as post-tissue samples. So collections of surviving corals, we were able to collect and sequence DNA both from the host corals as well as the symbionts from over 1,000 different individual corals, and this ranged across 3 keystone species and included 190 environmental samples to really get an idea of what's going on around corals in the environment when subjected to stress. And we chose 3 species, Acropora hyacinthus, Acropora millipora, and Acropora tenuis, not only because they were well distributed in our tissue storage samples, but also because they have a variety of different responses to bleaching. So tenuis is generally very resilient to bleaching. It's a hardy species. You seem to find it even after quite extreme environmental stress. And that compares to millipora and hyacinthus, which are generally much more vulnerable to bleaching stress. And then we really had no idea how the communities in the sediments or in the benthos would respond. So we used deep sequencing, and we sequenced across a number of platforms to really get a good idea of what's going on at the symbiont community, both the dominant players in the communities as well as the background low abundance symbionts that we know are really important for coral physiology. And if you think coral taxonomy is a nightmare, symbiont taxonomy is even worse. And there's hundreds, potentially thousands of different symbionts living inside of corals as well as free living in the environment, many of which are uncharacterized. And for today's talk, I'll just be focusing in on Durastinium, which is generally thought of as the stress-tolerant genera, the heat-tolerant genera. So I'll just be talking about them today. So when we looked at the prevalence of these different heat-tolerant symbionts, we actually found that they were quite low across the GBR before bleaching. So when I mean prevalence, I mean just the presence or the absence. So if we take this little example here, coral 1 and coral 3 had these symbionts, but the middle coral did not. And when we looked across all of our different biomes, we could see in our pre-bleaching samples that most of the colonies on the reef actually didn't have these heat-tolerant symbionts. And it was only after the 2016 event that these really started to pop up, and in particular in Acropora millipora, which we know to be quite a flexible coral in terms of its symbiont associations, and that interestingly, the sediment samples were just chalkers with heat-tolerant symbionts both before and after bleaching. And that, really this suggests that this 2016 bleaching event really had an impact on these particular communities inside of corals, which we should be thinking about. So we also wanted to look at their abundance, so not just which colonies had them. And again, taking our example here, abundance could be kind of a shuffling of different relative abundances of different taxa. And if we just take a subset of our reefs, looking at pre and post, we can see again with these heat-tolerant genera that there was a 40% increase in the abundance of these heat-tolerant individuals or taxa, but unfortunately a 50% decrease in the sediments. And much of this was driven by these inshore reefs. So Roger talked a bit about biodiversity being a really important indicator of ecosystem health, and so we also wanted to look at, well, how is diversity being impacted in these complex communities? And we found hundreds of different taxa within corals, and even more within the sediments, which we expected a bit. And there was a bunch of transfer between these two biomes, which is really important if we're thinking about the sediments providing a source of heat-tolerant symbionts for corals moving forward. And I'll just take a subset of different taxa, and each of these little dots represents a taxa. But essentially, what we could see is that, at least in these heat-tolerant symbionts, that in the corals, although their abundance was going up, their diversity was going down. And so if we looked at this kind of on a northern to central gradient, we could see that diversity within corals in these important symbionts were decreasing, and that, interestingly enough, they were increasing in the sediments. So in particular, we really need to think, are we losing potentially key symbiont species, especially in regions like up in the far north, which we know got really smashed by heat during these large-scale bleaching events. So switching and shuffling, we found that corals essentially like to shuffle their symbionts in response to stress. So they generally change the relative abundance of their symbionts, but in contrast, the free-living communities really had wholesale shifts. So they tended to switch, so completely change their communities in response to these big stress events. And in particular, the heat-tolerant community really seemed to be impacted by these changes. So heat exposure on reefs affects corals differently, and heat exposure can be measured by degree heating weeks. So how much accumulated heat is essentially on these reefs for a certain period of time. And there's been work by Terry Hughes to show that around 4 degree heating weeks, we get about a 45% decline in coral cover, and anything over 8 really starts to have severe impacts on mortality in terms of coral cover. And so we wanted to tie in the kind of reef level surveys that we were doing with symbiont communities with actually how these reefs responded in the field. So I wrote to Terry, and he provided his aerial survey scores, and we downloaded the satellite data, and we calculated essentially a deviance score of per reef how much heat stress was accumulating on these reefs, and then how did that reef respond in relation to that heat stress. So this was really to get at this idea of ecological memory. Was there a fingerprint of bleaching on these reefs that we know experienced a lot of heat stress? So here we have the bleaching scores, where 1 is not so bleached and 5 is a very bleached reef. And we have max degree heating weeks here on the Y, and we can see that we sampled reefs of only survivors. And that in particular, these were really interesting because these were reefs that got smashed by heat, but for some reason did not bleach. And alternatively, these were individual reefs that accumulated quite a bit of heat stress and did bleach. So we wanted to compare these So we took all of our thousands of samples, took our symbiont communities, and what we saw was that essentially symbiont communities in these reefs that accumulated a lot of stress and did bleach did not change before or after. So there was something going on with the host, some kind of intrinsic factor that was determining more their response to stress. So those were our most vulnerable reefs, those reefs that accumulated a lot of heat stress and responded. But what about the toughest reefs? What about these guys down here that accumulated a lot of heat but didn't seem to bleach? And so in particular, there were 4 reefs that fit this profile. And when we looked at our symbiont communities in our most flexible coral, Acropora millipora, as well as tenuis, our toughest coral, they tended to do the same thing in terms of their symbiont communities in that they didn't respond at all. It's almost as if the colonies didn't even register that there was a lot of heat going on. So that, again, suggests there's something intrinsic about the host, about the animal side of the equation, that is determining more their response to heat. So we took 200 of those millipora colonies and did full genome sequencing for them and correlated some of the environmental factors and found that the saxin gene was a particularly important gene in explaining a lot of this bleaching tolerance. So it's both the response of the reef is both a response of the symbionts as well as the host. And so why should we really care about these heat-tolerant symbionts that are free-living outside of the corals? Well, on the Great Barrier Reef, about 85% of coral species undergo mass spawning during October, November, and a majority of those need those free-living symbionts in order to recolonize the baby corals at some stage of their life cycle. So it was really interesting in that 2016, there was really a very low preponderance of these heat-tolerant symbionts, although they were very available for corals to take up. And it wasn't till after this bleaching event that we really saw the increase in abundance in corals afterwards. So looking forward, we know that these kind of monitoring studies, that they're observations and that, correlation does not equal causation, but it's a really important part of, understanding and observing something and then doing some kind of manipulative experiment and then to understand the mechanism. And we can do that at a place like the National Sea Simulator at Ames. And we actually have run an experiment looking at coral and juvenile uptake of these different symbionts, and we found that in future warmer conditions, that baby corals did preferentially take up these more heat-tolerant symbiont types. And when we think about diversity on the Great Barrier Reef, we talk a lot about protecting kind of megafauna, but potentially we should also be thinking about, protecting symbioses as well. And so as we kind of went along on this project, we started to accumulate quite a lot of genomic data in a variety of forms. And so that kind of led to a number of discussions with Grabumpa on how we could start to translate all this genomic data, transcriptomic data, into more solid management actions. So we're currently writing a paper, a blueprint about this, and next week we'll meet with Grabumpa again. They'll have a look and we'll see what they think about this blueprint. And also a part of this NEST project, we had the opportunity to speak with a lot of traditional owners before we went into sea country to essentially look for samples. And during this time, there was a lot of conversation about the use of genetic material from sea country in these kind of management scenarios. And so that has really led to some further work now with the National Geographic to kind of formalize some of these conversations. And that wouldn't have come about without NESP. So some of the outcomes, there's been a lot of learning about how to incorporate traditional knowledge into reef research from the beginning, not in the middle, not at the end. We've been able to identify a number of reefs at risk, resilient reefs, as well as at least start a roadmap for translating some of this genetic information into adaptive management. So with that, I'd just like to say thank you to everybody at NESP and everybody at Ames and the Legacy team. Thanks.